Thanks for joining. Can everybody see my presentation correctly? All right, great. So today we'll be giving you a bit of a whirlwind tour of some modeling techniques with the idea of just giving you a taster of how we think about integrating models with experiments across organizational scales. So this will be with me and Silverman and Katie Knaus at Colorado School of Mines. And one of the reasons we want to touch on this is because one of the three important integrative themes across the IMSI network is bridging hierarchies with multi-scale modeling. And earlier you have already learned about a couple of modeling approaches at the lower organizational scales, thinking about molecular mechanisms of muscle contraction and muscle contractile dynamics, and then all the way at the sort of higher end of the scale with Christian Hubicki's optimal control introduction, where we can think about, use these kinds of simulations to think about how organisms interact with their environment under different conditions. So we're going to be touching on some modeling approaches that span in the intermediate range of these scales. So if you haven't already, if you want to join in the interactive components today, please go ahead and make sure that you have downloaded the Google Drive folder that's linked in the Slack general channel, and make sure that you have the MATLAB files as well as the OpenSim files on your computer to help go through these. So what we'll be doing is talking about three specific scales of biomechanical models. I'll start by kicking off with one of the simplest models of locomotion that's possible, the mass spring model, which represents whole body dynamics at a very abstract level. And we'll be talking about musculoskeletal modeling of whole body dynamics. And we'll talk about how we might integrate between these different approaches a little bit. And then Katie will be talking about finite element modeling of muscle tendon dynamics. And one of the themes that we want to touch on throughout this workshop today is to think about how do we effectively integrate our models and experiments. All models are simplifications, and so we have to make assumptions and estimate parameters. And so all models are wrong in some way, and we want to think about what can we say and what can't we say based on a particular model at a particular organizational level. Oops, sorry. So some of the key questions we want you to think about throughout the session is what biomechanical data are needed to drive each modeling approach? What are the key inputs and outputs from each type of model? And what data would we need to validate each model? So I'll be talking briefly about template models. So template, when I say template models, this is a very abstract model that represents the task level mechanics in a way that really generalizes across diverse organisms. So it doesn't include the detailed features of the organism's anatomy. And the most famous and commonly used example of that is the mass spring model. It's also, when we use the model to simulate forward locomotion, it has both the bouncing mass spring element and an inverted pendular motion. So it's called the spring-loaded inverted pendulum model. And this has been used as a template model for legged gaits. And what's interesting about this model is that if you abstract the body as a point mass at the center of mass, and you extract the legs as a single virtual leg that exerts force against the ground in proportion to its compression, so acting as a linear spring, you can simulate bouncing legged locomotion. And despite the diversity of morphology, number of legs, and locomotor styles you see among animals, this model can accurately represent the body motion and the ground reaction force patterns observed in fast gaits across diverse animals. So this model can simulate reasonable approximation of the magnitude and the timing of ground reaction forces for animals as diverse as hopping kangaroo rats or hopping wallabies, ostriches and other running birds, humans, quadrupeds, six-legged animals like cockroaches, and even eight-legged animals like crabs who are moving sideways. So when we think about this model, this is very abstracted. So what are the model inputs in this case? In this particular model, right? We need to have some very basic information about the scale of the organism. So you would have body mass, you need to know the body mass and an effective leg length for that animal. The output of the model in a simulation would be the predicted body motions and the predicted ground reaction forces for a gait. And how might we validate this model? So I'll be giving some examples to show how we might use this model to explain diversity of locomotion in organisms and consider some of the factors that differ between organisms. But one of the ways we can validate this model is by directly comparing observed body motions and ground reaction forces from experimental data to the model predictions. And I'll be talking a little bit more about this a little bit later on. Okay. So template models are very abstract. They're in fact clearly wrong. You know, we know that the legs are not massless springs, right? But they still provide a good representation of the whole body dynamics and a prediction of the ground reaction forces. And so they can explain the general gait features and model how the ground reaction forces relate to body motion across a range of gaits, from walking to running, and changes with speed, different stride length and stride frequency patterns. But to really understand any musculoskeletal mechanisms, we need more anchored neuromechanical models that actually include some of the actual detail about the musculoskeletal anatomy and the muscles that actuate the system. And so that's what we want to talk a little bit about today, is how we think about integrating across these scales and modeling approaches, because that's one of the big goals of our Integration Institute. So what is a simple model good for? It can't tell you anything about the actual underlying neuromechanical mechanisms that lead to the observed motions and ground reaction forces. So it's not good for that, but it can be used to understand features of gait and potential mechanical trade-offs in locomotor behavior. So I'll try to give some examples of how that can be used later on. Okay. So why do we need this model? One of the things, it seems very simple, but in fact, it's useful because despite the apparent simplicity of bouncing gaits across organisms, it's actually pretty complicated to try to predict what the ground reaction forces will look like for a given motion. Because legs are not wheels, right? If we were in a car, we would have very consistent forces supporting body weight with very few fluctuations, right? But locomotion involves intermittent contact on legs, where there's decelerations and accelerations with each stride, and fluctuations in mechanical energy that depend on the timing of contact of the limbs with the ground, because the limbs are exerting the force against the ground that helps to resist gravity and support body weight. And so you can think about this as a hybrid dynamic system. So when we think about legged locomotion, the equations of motion of the system are not constant throughout locomotion, because the actual dynamics and the equation of motion depend on the phase of gait and how many legs are in contact with the ground, right? So if we're just thinking about a simple biped or hopping locomotion, you have a stance phase where a limb is in contact with the ground and generating force. And then you have an aerial phase or a flight phase in running, where the body is just falling with the only force falling with gravity, right? So it's just ballistic falling. So the dynamics of the system depend on whether there's 0, 1, or more legs in contact with the ground. And the mass spring models can help us understand how the ground reaction forces patterns differ depending on the timing of ground contact and the duration of the swing or the aerial phase and the stance phases. All right. So if we think about experimental observations of ground reaction force, this is a video from Jill McGray, who's also in our network, of a runner running across a force platform that's in the middle of the runway. And you can see the ground reaction force trace that shows the vertical and the fore-aft horizontal components of the ground reaction force that correspond with that gait. And you can see there's a little line that goes across when it hits the specific plate that's recording the force to show the timing of the force trace, the force development relative to foot contact. Okay. So there are characteristic features that have been experimentally observed, right? The vertical component of the ground reaction force is always the largest. In running, when there's an aerial phase, it rises to a peak and has sort of a half sine shape, right? And the fore-aft horizontal, there's a decelerating impulse in the first half of stance and an accelerating impulse in the second half of stance. And these features are very characteristic across organisms. So we can compare to an ostrich, despite having a completely different morphology. It has fairly similar characteristic features of the ground reaction force. Now this ostrich, it's not running perfectly steadily, so you can see that there's some variation in the peak magnitudes and the timing, but the general shape of the curve is that there's a large vertical impulse and then a decelerating and accelerating fore-aft impulse during each stance phase. Okay? And so the mass spring model can be used to represent the magnitude and timing of these ground reaction forces. If we look at ground reaction forces for bipedal walking and running across bipedal species, so species as diverse as humans and ostriches, they still have characteristic patterns in how those forces change with speed and gait. So during walking, you get a characteristic M-shaped curve in the vertical direction, and this decelerating and accelerating impulse in the fore-aft direction. In running, you get the single peak during the stance phase in the vertical direction and a similar fore-aft horizontal impulse. And as speed increases and the duration of contact decreases, peak forces rise. So the magnitude of the peak force is related to the duration of contact. And you see that as we go from slower running to faster running, there's an increase in the magnitude of the fore-aft impulses as well. So the mass spring model can be used to predict the specific magnitude and timing of the ground reaction forces for a particular gait. So if we think about, so we have a point mass bouncing on a massless leg spring, and if we simulate this, which we'll do in a minute in our interactive activity, you can predict the ground reaction forces. And of course, the model is so simple, it can't explain all of the features of the ground reaction forces, but this is an example of modeling the ground reaction forces of a guinea fowl running in this case. And you can see that there's the magnitude of the vertical force and the magnitude of the decelerating and accelerating impulses are accurately represented by the model. But the shape of the ground reaction force curve is much simpler for the mass spring model compared to real ground reaction forces, in part because real animals have legs with masses that result in collisions when they contact the ground. And so you get heel strike impacts associated with these collisional decelerations of the masses in the foot and the leg. Okay. But basically, the mass spring model tells us how the ground reaction force magnitudes change for a specific pattern for speed and cadence of a gait, right? Step length, step frequency combination. When we just use basic Newton's laws to ensure that average vertical forces support body weight over the course of a steady gait cycle, and the horizontal fore-aft forces average 0 over a steady gait cycle to maintain a steady speed, right? So for steady running, we can constrain our model so that it starts and ends at the same speed and the same total mechanical energy, and then we can observe the ground reaction forces that are required in order to maintain body weight support and that forward speed. All right, so I'm just going to walk through the equations of motion of the mass spring model really quickly because that's what is required to run the simulation. And then we're going to run an example simulation of mass spring model in MATLAB. And so the simplest version of the mass spring model is actually a hopper, a model of vertical hopping. So I just want to walk through that. Basically, where we have a point mass for the body, massless leg with a stiffness that we approximate as an effective linear leg stiffness, K leg, and it has a leg length, which is the distance from the center of mass to the ground, okay? And so if we just apply Newton's laws, F equals ma. The sum of the forces equals ma. We have gravity acting in the downward direction, right? And then the ground reaction force exerted by the leg opposes that. So if we write this out, we have the sum of the forces in the vertical direction only, because that's the only thing we're considering here. Fg minus mg equals ma. And in this case, we're approximating the limb as a simple linear spring. So the ground reaction force is proportional to the stiffness of the leg spring times the compression of that leg spring. So that delta L, the compression of the leg spring, is calculated as the L naught, the resting length, right, which we would measure as the standing hip height of an animal, minus L, which is equal to the motion in the negative x direction. Okay. In this case, we just have a single direction of motion, which is the vertical. So negative x is the vertical motion. So we now can combine these two equations, right, and substitute in k leg times delta L for Fg in the first equation, right? And we get this, k leg times delta L minus mg is equal to ma. And we can rearrange and solve for acceleration. Okay, so if we solve for acceleration, by rearrange these, dividing by body mass, you get the equation of motion for this hopping system. The acceleration is k leg over m times delta L minus g. Now, the trick with the spring-loaded inverted pendulum is that there's also forward motion in the system, right? And so we have to adjust the equation of motions to account for the fact that the system has motion in both the vertical and the fore-aft directions, okay? So for our slip model, okay, it's still the same physical system, but now the leg contacts the ground with an angle relative to the horizontal that is not 90 degrees, right? And we have to, since gravity is only acting in the vertical direction, we have a different equation of motion for the fore, aft, and the vertical directions. So in this case, I've defined my axes so that x is forward and y is the vertical direction. So our equations of motion for the slip model are, we have to account for the angle of contact when the foot hits the ground, okay? So we have acceleration in the x direction, that's forward direction, gravity is not acting. So we just have k leg over m times delta L times cosine of theta. And then in the vertical direction, we do have gravity acting. So it's k leg over m times delta L sine theta minus g. And those are the equations of motion that we're using for the simulation that we're running today. Okay, so we're going to go ahead and switch over to MATLAB. And so make sure that you have MATLAB running and run an example simulation to show how you can simulate different gates using the mass spring model. So I put these instructions also in Slack. You should make sure that you have the mass spring simulation folder downloaded. Open MATLAB. I'm going to just switch to MATLAB and do these steps so that you can see. So I'm going to just exit my screen share, stop sharing here, and share my other screen. Can everybody see my MATLAB screen now? Okay. So once you open MATLAB, okay, you should be able to see your command window and also the file browser. Make sure that if you've downloaded the files for the simulation, you go and browse in the file browser to the mass spring simulation folder, and you can then, so that you can see the files like I have them here, okay? And then also go to set path and say add with subfolders and add this folder so that in your set path browser, you can see the simulation files in there. So I'm going to save and close that. Once you've done that, and I'll pause to let people check that theirs is working, there's a little test function to make sure that the simulation files are configured correctly in MATLAB. So you can say test ball in box, and it basically runs a simulation in the background and tells you if it simulated that correctly. So you should be able to get this success message if you've done that correctly. So I'm going to pause for a minute and let people try to make sure you have time to do that. And I'm also happy to take a couple questions while people start getting that ready. Are people able to do that? Anybody have questions or problems?
It's working well on my end, at least. That's all I can attest to at the moment. That's good. I have a
Okay.
bouncing ball. Did I cheat? I have a bouncing ball.
Oh yeah, if you run the ball and box simulation, you'll get so there's a example ball and box that will actually show you the simulation. Oops, I typed it incorrectly. And so you might get a bouncing ball in a box if you actually run that simulation. So
I
this
do.
is just testing the hybrid dynamic simulation framework, that it's running correctly. And the reason we have a hybrid dynamic simulation framework is that we have to be able to switch between stance phases and aerial phases in simulating our mass spring model, okay? And so a complete cycle is an aerial phase so we start the simulation with the model at its apex height during a flight phase, and it falls until it contacts the ground, and then we have a stance phase, and then it bounces up to the next apex. So we have a falling phase, stance phase, and then a rising phase. Okay, so if you manage to get the simulation working, then you should open, if you go to the basic slip folder, open your example mass spring runner file. And can everybody see the actual code now on my screen? Okay. So I'm not going to walk through all the details of the code, but if anybody's interested, you can come talk to me about it afterwards. So this is basically, it has a framework to set some default initial conditions and parameters that you need for your model, and that's what we're going to play around with. It then runs the simulation for one gate cycle. It finds a steady state solution, and then it plots the results, and we'll see the results. So open the get default ICs and params function. So if we open that, then you need to actually go to line 42. Okay, so in this function, lines 41 through 46 are setting the parameters of our model. We have a leg angle, gravity, the body mass in kilograms, the resting length of the spring, we just set it to 1 here, and we have a spring stiffness. The spring stiffness, we're going to start by setting it to 5,000. That's actually very compliant stiffness in this case. So these parameters, 80 kilograms and 1 meter, is approximately sort of a human scaled simulation of a mass spring runner. And we're going to set the stiffness to 5,000. And now if you go to line 98 or thereabouts, okay, we're going to set an initial bounce height guess. So we need initial conditions, and one of those initial conditions is the bounce height. And this is just to give us a value that's pretty close to our steady state solution so that the simulation can find the steady state solution easier. We're going to change that, make that 0.75. And if you save that, then go and run your example mass spring runner script. And now I don't have anything happening yet. It did run. Where did my movie go? It should produce a movie. Has anybody got a movie in theirs? Did you get it to run with a movie? I think it might be hiding on someplace on my screen that I'm not seeing. So that's, okay, hold on just a second. Movie when you ran yours, please.
We got us to run.
We got it at 10. Looks cool.
Okay, so you got a simulation to run.
Yeah.
Okay. I don't know why mine's hiding from me right now, but let's see. Okay, I can show you if we go back to my PowerPoint. You should get a movie. I can't find mine right now because of technical issues, but you should also see some information printed in the command window about the resulting gait. So it tells you the peak force in body weights, the peak fore-aft force, the normalized velocity. We're not going to get into the details of that. The velocity in meters per second, bounce height, and some other things like the stance period and swing period. But I'm going to show you what it looks like in terms of the the movie that I created in mine, so you can, I created earlier. Okay. Okay. So if you change the parameters, you should get a movie that looks like this. See where you can see a graph of the ground reaction forces that are modeled. It shows the vertical, fore-aft, and the vector magnitude of the forces. And you can see that the gate has a very long stance phase, right? And a relatively short aerial phase. So this compliant gate has very low bounce height and a relatively smooth motion, right? A relatively long distance traveled during the stance phase. So now we're going to go back to our code and change it one more time. Okay, so if we go back to our initial conditions in parameters code, sorry. Now, I'd like you to change stiffness by adding a 0, so change it to 50,000. So it's 10-fold increase in stiffness. We're keeping all of the other parameters the same here, except I know that this is a high stiffness results in a very bouncy runner. And so we're also going to increase the bounce height so that it gets us a plausible initial condition here. So we're changing the bounce height to 1 on line 98. Yeah.
We can't see your screen.
Okay, I'm sorry. I forgot to start the sharing on the MATLAB screen again. Thank you. Okay, so going back to the MATLAB. Okay, all right, so we're going to up on line 46, add a 0 to stiffness to change it to 50,000. And on line 98, we're just going to change our initial guess for the bounce height to 1. So that's about 1 leg length. Higher stiffness ends up resulting in bouncier, vertically bouncier gates. So we need a little bit higher height. Now you save that and go back and run your simulation again from the example mass spring runner script. And my movie is not showing up for some reason. Are you getting a movie? Anybody?
We got a movie.
We were able to get it.
Yes. Okay.
Yes.
So I can tell my simulation ran because it printed new results for the limit cycle that was found with a higher leg stiffness. So remember, this is the same model with the same, approximately the same forward speed. The limit cycles may vary a little bit in the exact forward speed. They have the same body mass, the same leg length, and all of we've changed is the effective leg stiffness. So this is the same runner running with a very compliant leg or a very stiff leg. And you can see that the peak forces here are almost 4 body weights, right? So we have something that's going to have a higher peak magnitude of force. So I'm going to go back to my PowerPoint again, switching back.
Hey, I think you might be able to see the figures you're creating if you right-click on the MATLAB icon and then click on the figure number.
Oh yeah,
I go back to PowerPoint.
Well, I'm going to have it in my PowerPoint anyway. So I pre-generated them in my PowerPoint. So here's the movie. Can you see that? So basically need to confirm that you got a similar solution when you ran your simulation, right? Anybody? Yeah?
Yes, correct.
Okay. So this is a pretty simple example, but basically it's demonstrating that a runner running with approximately the same forward velocity, there's different styles of gait, right? You can run with a more compliant gait or a stiffer legged gait. And this does change the gait parameters, including the bounciness of the vertical motion, right? So this stiffer legged runner has much longer aerial phases, much higher peak vertical forces associated with this gait, okay? So how can we actually use this in the context of data? So I know I'm running a little bit over time. Apologies to Anne and Katie. We'll give you your time too. We use this model in the context of comparing bipedal gaits across running birds from quail up to ostriches by looking at their actual gait patterns experimentally and matching their stride patterns using this mass spring model. So getting stride frequency and stride length patterns at a given speed that matched the empirically observed data, right? So you can look at these videos of a quail compared to an ostrich, and you might be able to observe just qualitatively that they do have different running styles. Smaller birds tend to have more crouched postures and more compliant gaits with smoother vertical motion, whereas larger birds tend to have a more vertically bouncy motion of the center of mass, okay? And we compared gaits across a wide range of animals, so we compiled data from the literature across 21 species, and you can see that there's a general trend towards relatively higher stride frequencies and relatively lower stride lengths for their size in large animals compared to small animals. So there are different gait styles preferred by small versus large animals. And we can use the mass spring model to say, okay, given these gait parameters, if we try to match the gait parameters in mass spring model, what does this predict about the features of the ground reaction forces for these different animals? So if you do this, you find you can simulate mass spring dynamics. I'm not going to show that in detail, but qualitatively what the comparison is shows that small animals have relatively higher fore-aft forces because they take longer stance phases, so they have a more compliant gait with a longer stance duration. They have higher stance leg work if you consider the work that's loaded into the spring through the compression of the spring. In reality, that's done by muscles, right? Or some combination of muscle and tendon. They have, but they have a higher terrain robustness, which we quantified by what's the biggest magnitude of a terrain drop that they, in proportion to their leg length, that they can handle before they fall. And they have lower swing costs because they have a longer duration for the swing period of the gait. In comparison, larger animals have lower forces, fore-aft forces, lower stance leg work, but lower terrain robustness and higher swing costs. And this might make sense in the context of potential ecological trade-offs or biomechanical trade-offs faced by small versus large animals, where an ostrich probably doesn't have to navigate obstacles that are like, 50% or more of its leg length, but a quail probably does that fairly regularly. So they have to, for their size, be able to navigate relatively rougher terrain. What's interesting is if you use the mass spring model kind of framework to compare gaits across species, the gait of an ostrich is in some ways more similar to the human than it is to smaller birds like quail or guinea fowl. And so there are some scaling effects that seem to be important despite the very different morphologies between humans and ostriches. And I'm going to stop there. I had one more example, but I know I'm over time. So I'm going to go ahead and skip this example, and anybody who's interested in it can ask me about it, but I'm going to go to sort of discussion points here. Thinking about what are mass spring models good for, or similar kinds of template models. They can influence how gait parameters influence ground reaction force magnitude and timing, and they can relate whole body motion to ground reaction force patterns. So you can use it to investigate things like how hole and stiffness influences gait dynamics. We have also used it in the context of understanding how perturbed landing conditions influence gait dynamics in unsteady gait. And there's a paper in the shared folder that you can read if you're interested in that. They're really terrible for other things, right? You can't replicate all of the details of ground reaction forces like the heel strike impact or asymmetries. And you can't really identify specific mechanisms of musculoskeletal mechanics or sensory motor control because it's abstracted at such a simplified level. Okay, so with that, I'm going to pass the baton over to Anne. Apologies for going a little bit long.
All right, thank you. Go ahead and share my slides. Everybody see that? Great. So moving down the scale or potentially increasing complexity with still some limitations. That's sort of the next phase that we're talking about in this whirlwind tour of models. I have a question for you, which is that, why would we want to model and simulate movement at this musculoskeletal level, which we're going to talk about for a few minutes here. And so these two questions are kind of linking from Monica's last slide and are sort of the same question as, there are some limitations to spring mass models. Why would we want to know other things, right? Or what would we use musculoskeletal models for, which have some greater complexity, still have some limitations, and you know, what's the reasoning behind this? Well, I think there's a lot, and this is certainly not a comprehensive list. But I think a big thing with IMSI is how do we apply principles of movement coordination across species, which certainly spring mass models can do as well. But if we're looking at things at the muscle and joint level, particularly internal to the body, we can't measure those things directly. We can't instantaneously and continuously measure muscle force, at least not currently, and certainly not from all the muscles in our body. So these models allow us to systematically change different parameters, explore theoretical scenarios, and that can lead to comparisons across species, understanding movement disorders, designing assistive devices, and of course, linking with neuroscience principles like we talked about at the workshop on Tuesday. So there's lots of reasons to use these types of models, but we need to understand what input data we need in order to make them run effectively or to understand our results in context. So my question for you is, what data might we need that could help us simulate and model motion? And this is a very generalized question, so there aren't really wrong answers. If anyone wants to unmute or maybe put something in the chat, think big. Thank you, Christian Hubicki. Kinematic data. Leah Houston, timing of muscle activation. You both are absolutely right.
Muscle
Motion
properties.
capture data.
Muscle properties. And I heard something else. Say it again.
Motion capture data.
Motion capture data, absolutely. So you guys are all correct. I think in modeling studies, I think my answer is absolutely everything we can possibly measure is going to help us. But one of the challenges with linking modeling and experimental studies is that we can't always measure everything, or it's not feasible, or it's not practical. But some big sort of categories that you all touched on, and what we're going to talk about today is movement data of some kind, optical motion capture, inertial measurement units. I know there's a lot of projects in IMSI looking at videos and how we can extract movement from that. External forces are a huge one. So those ground reaction forces that we just predicted, you know, knowing what those are can really help us. And then of course, muscle data, muscle properties, also real-time sort of movement muscle information, such as electromyography or fiber velocities, geometry of muscle from images can be really useful. This is certainly not a comprehensive list. So examples here of kinematic data. This is just human walking data, sagittal plane kinematics, hip flexion extension, knee flexion extension, ankle flexion extension. We can measure those with a motion capture system. Ground reaction forces, very similar to what Monica just presented. You know, during walking, we have the vector representation in the big picture on the left, as well as the 3D components in the fore aft direction or anterior posterior, the medial outer direction, side to side, as well as vertical. And so knowing how an organism, human or animal, that is interacting with environment is really important to understanding the dynamics of the task. And then of course, muscle data. We collect a lot of this in our lab in order to validate a lot of our muscle solutions from musculoskeletal simulations. Of course, we can't measure this from every single muscle in our bodies, but knowing this information can help us understand our solutions better, where they might be falling short or where they might be more realistic. So linking back to those questions from the beginning is, what do we use for input? What do we use for output? What do we use for validation? I think these are questions that we really need to ask it that can vary depending on your question. So there's not just one answer here for musculoskeletal models. We might use different data for input, different data for output, and validation is an extremely important part of that overall process. And so one of the things that indicates what data we use for input versus output is the approach we're using to developing a musculoskeletal simulation. So this workflow here is a very common description of how we can run forward or inverse simulations. So if we start at the left, we've got this neural command from our brains, and this is how we produce movements as animals, right? We start with some neural command, and then we need to understand this excitation to muscle force relationship. And we've had a lot of great workshops about how can we do this well, right? In terms of what underlying muscle model we use. Do we use a hill type model? Do we incorporate different elements? Do we go even with greater complexity, like Katie Knaus is going to talk about in a second? So in this case, and the activity we're going to do today, we're using a simple hill type model, which of course includes our force, length, and velocity properties that we've talked a lot about, in order to make this bridge between excitation of a muscle and what forces are actually produced. And then we have a lot of geometry. Where do those muscles attached? In the model we're working with today, we have simplified the muscle geometry to single line, one-dimensional geometric representations with an origin and insertion. Maybe they have a wrapping surface, like you can see here in this sketch. And those linear simplified force actuators are then applied to a dynamic model that includes center of mass information, inertial properties, length, leg lengths, things that all vary a lot across individuals. And we then determine our equations. We apply our equations of motion in order to determine that moment to acceleration relationship. So this is all Newton's laws in here. And then we can integrate in time to get velocities and positions and an output movement. And there's certainly sensory motor control, as James Finley and Kelly Sharp talked about on Tuesday, that can then adapt this neural command to address changes in our movement over time. So when we're running a simulation, a forward simulation, we start with this neural command and we move it forward. We do each of these steps to get our output movement. Another approach is an inverse approach where we start with an observed movement, and this is often from experimental measurements from a lab. We differentiate instead of integrate, and we follow these exact same steps, but move backwards. And one of the challenges with such an inverse approach is that we have way more muscles than degrees of freedom, which means that we have way more unknowns than we have equations, which means that we need to apply optimization techniques like Christian Hubicki talked about in order to determine what muscles are doing what when in order to produce the movement that we observed. So in terms of developing one of these analyses, let's go ahead and do this together. So what we're going to do today, there is a subfolder in the download specifically for a static optimization activity in OpenSim. So launch OpenSim if you haven't done so already. That folder has all the input files you're going to need. It also has a Word document with the step-by-step instructions that we're going to do together. We're going to run 3 scenarios, baseline hopping, and this is hopping data from our lab here at Colorado School of Mines. We're going to do another simulation where we've changed the strength of certain muscles, and then we are going to look at a different objective function and compare what these solutions look like when we're trying to figure out what these individual muscles are doing. And there are output files and reference plots that are included in this folder as well, in case something doesn't quite work out as expected. You have those to compare and probe and go more deeply into. So let's go ahead and I will stop sharing my slides and I will share my OpenSim screen, and hopefully, I don't have quite the wizarding skills that Dr. Hubicki does, as he showed us last week. Let's see if that works. All right, I think everybody should be able to see OpenSim. Is that correct? All right, I see a thumbs up. Okay, so launch OpenSim yourself. You should see the same screen. What I'd like for you to do is go to File, Open Model. So we're going to start with a model that's been provided to you. And it's going to be in this main folder. There are many files. We're going to start with this participant 1 size scaled gate 2392. Now, Gate 2392 is a generic model that comes with the OpenSim download that you should have on your computer. This one has been scaled in size and body mass to the participant in our lab. What the 23 means is we have 23 degrees of freedom and 92 musculotendon hill type actuators. I'm going to go ahead and open this. And hopefully you should see it on your screen and mine. And if we go over here to this navigator tab, you can expand that says participant 1. You can see we have a lot of different components in this model. We have ground, we have bodies, which means, if we expand this, this is all of our body segments or bones in terms of our mass and inertial properties. And we've got our joints, how they're connected to one another and how they can move. We also have 92 musculotendin actuators deep in this muscle list here that are currently by group. So there's a lot in this model, and you can open up this file in a text editor and see the thousands of lines of code where we've defined a lot of information about the personal characteristics of this particular model, which is usually based on a subset of cadaver studies, and may do a great job at representing you and me, or may do a terrible job at representing you or me in terms of our body characteristics and our muscle capabilities. We go over here to the coordinates tab. You can see this is all of our degrees of freedom. So I'll go ahead and go to hip flexion right here. You can move these around and see this model moving around like we want it to in different degrees of freedom. The next thing I want you to do is go to File, Load Motion. And we're going to go within this folder. There's a subfolder called Input Files. It has 2 files in it, one labeled kinematics and one labeled ground reaction forces. Make sure that you select kinematics.mot. Go ahead and open that. And if you go here to the top of OpenSim, click this looping button to the right of the time bar and press play. You can see I've put my speed at 0.5, so you might consider moving your speed down a bit so you can visualize it. But here we've got double leg hopping. So this is real data that we collected in our lab. There's quite a bit of processing that goes from those marker trajectories from optimal motion capture and applying it to a model with specific degrees of freedom. But you can see this person's bouncing up and down, hitting the ground, flexing their knees and hips, and extending up into the air. And so what we want to know with this exercise is what muscles are causing this motion to happen. We haven't figured that out yet. All I've done is apply a motion to this model and see it move, but there's no force actuation quite yet in terms of how the muscles made this movement happen. Let's go ahead and pause that. Now, if we go to Tools and Static Optimization. All right. Quick check, is everybody caught up? A few thumbs up or yeses in the chat. Do we see what we need to see? All right. Thank you. All right. So we've opened the static optimization tool. So this is, like I said, we have way more muscles than we have degrees of freedom. So knowing what muscle's doing what when requires an optimization technique. I want you to go down here to load and go back up to your main working folder, and there should be static optimization _ setup.xml. So this is, I have set up some settings for you. Should be able to easy run, and let's talk through them briefly here. So how we set this up. First, we need to say, okay, here's the motion that I want. And you'll notice this file right here, participant 1 kinematics. This is the same one that you just loaded and animated. And so we have a motion that we're trying to analyze. We want to figure out what the muscles are doing. Next thing is we go down here to our objective function. Again, call out to Christian Hubicki's optimal control workshop last week. So the objective function we're going to start with is sum of muscle activation to the power 2 or squared. And we want to check this box that says use muscle force length velocity properties. So that's using those force length velocity properties of our hill type model. We have a certain time range of our hop. And then down here, we have our output directory. So we're going to change this one later, but keep it the same for now, static optimization results. And this is where all of our output files from this analysis are going to live. So we want to keep that organized and know exactly where it is. Next thing, if we go to actuators and external loads, I've added an additional set of forces, which are idealized torque actuators, which we ideally want to be 0. But basically, this is a fail-safe in case our muscles aren't strong enough, we'll still get our simulation to run. So that should be set for you. And then this external loads box is really important. What external loads do we have on this model, or during this motion, I should say? Chat or unmute. We definitely have gravity, and that has been defined. What else? So if you walk into, or stand up right now even, and jump up and down a few times, what forces are acting on your body besides gravity? Ground reaction forces, exactly. The ones that Monica just talked about, right? So if I don't define these ground reaction forces, that means this model is just going to fall straight through down into the abyss, down to the center of the earth. So we want to make sure we define those external loads. And so I have a folder defined here, external forces. I'll go to edit so you can see the components. It starts with this file that was in your input files folder. It says participant 1 hop GRF. And so these are also directly measured from force plates in our laboratory. And what I've done here is define the right one to the right foot and the left one to the left foot. And I've defined those 3 components as well as the point at which they're applied. So we have a motion, we have some forces, we can apply our equations of motion to figure out what our net joint moments are. And then we also have this optimization defined so that we can resolve those joint moments into what all those individual 92 muscles are doing. So we should be good to go. Go ahead and click run here and I will close this and you can see, hey, it's going. And you should see this hopping motion going a bit slower and these muscles are turning red. If they're completely blue, they're off. If they're completely red, they're at 100% activation. And you can see all of them are coordinating together in order to produce this hopping motion, which is a big reason we would want this whole body model with all of these individual muscles defined. All right, now we're going to plot some of these results real quick before I pass it over to Katie. But before we do that, I would like to run our other 2 scenarios and then we can plot them all together. So the next thing to do is let's open a different model. So now back in your folder, we started with this size scaled gate 2392. Now we're going to open size scaled doubled left gastroc strength. So go ahead and open this model. So this is the exact same model, except I have doubled the strength of the medial and lateral left gastrocnemius. Now, that's a little ridiculous, right? Can someone really double their gastroc strength and nothing else in their body? Probably not. But I do think this is really important in linking to some of our themes of IMSI, in that I have different muscle strength than you, and we certainly have different muscle strength in our quads versus our calves versus our back muscles. And so there's a lot we need to understand about how muscle strength might affect our movement patterns or how that might affect our muscle recruitment. And I think this fact that I'm able to do a ridiculous scenario is an advantage of modeling, and that we can change one thing in isolation just to explore how that might affect things from a systematic perspective. So we have a new model now, and we're going to do the exact same thing again. Go to Tools, Static Optimization. Let's load our static optimization setup file, open. Everything's the same. We've got the same external forces. We've got the same motion, but let's change where this output file is. So instead of static optimization results, let's name it something different. For me, I have picked results doubled left gastroc, something that makes sense to you in terms of that this is a different set of results. So I'm going to go ahead and do that. Everything else is the same. Press run and close. And you can see we're going through the exact same process again with those muscles being recruited, but now those muscles have different properties. And now let's do it one more time. We're going to use the same model now, Tools, Static Optimization. I'm going to load my settings, main folder, setup file. And I'm going to, instead of sum of muscle activations to the power 2, let's change it to the power 5. So basically what this does is it really heavily penalizes high activations of muscles. And let's change our output directly again. Results changed objective function. So everything else is the same. We still have that strong model, and we still have the same motion and the same ground reaction forces, but we're changing our objective function. Let's go ahead and run that and close. Okay. So we've now run 3 cases. Let's look at our muscle results. Now, the plot tool in OpenSim, not ideal for most cases, but I think it does work well in the fact that we're all doing this together and everyone has this program over and we can plot these results quickly. So if we go to Tools, Plot, right at the top, And let's make sure, can everyone see a plot on my screen that doesn't have anything on it yet?
No data yet, but we see the plot.
Yeah, okay. So let's plot these muscle activations for a couple different scenarios. If we go to Y quantity and let's load a file. So we've got a lot of different results here. So this is our, go to your static optimization underscore results. This is our first run, our baseline model. Go ahead and select participant 1, static optimization, activation. So this is activations of everything in our model. And when you press open, you should get a long list of muscles, all the muscles in our model. Scroll down. This is not, that's why I say what I mean, the plot tool is not ideal, but I'm going to focus on those left ankle plantar flexors. So we've got our medial gastrocnemius, our lateral gastrocnemius, and our soleus. Press OK. And then for our X quantity, we want to have that over time. So this is over time of our hopping simulation. Press add. And here we go. We've got 3 traces, our 2 compartments of the gastrocnemius as well as the soleus. So what do you guys notice about this plot?
It's like Devil's Tower.
Like it. So basically we've got this flat line up here at 1. That's the max of our activation, right? For all 3 muscles. So all 3 of these muscles are maxing out. And hopping, double leg hopping is certainly a challenging task, but I don't know if I would call it maximum. It's more of a jog as opposed to a sprint. And so what this makes me wonder is, okay, is our model just not strong enough? Is that really reflecting our participant and their capability for this particular model? Now, if I go to, you'll remember we ran one with our strong gastroc, so we only strengthened 1 muscle group, but we did make it stronger. If we plot the same thing again, so we can compare these right on top of each other, we've got our medial lateral soleus. Press OK as a function of time. Let's go ahead and add that. Now, I realize it's hard to see with 6 traces on the screen, but the main thing, if we compare our soleus, the green to the black, you can see not maxed out anymore. And the gastrocs are still maxed out, but for a much lower duration. So we're moving in the right direction here. If we go ahead and I'm going to remove our baseline just to clean this up a little bit. Here's our strength, our scaled model, or our increased gastroc strength model. Now, let's look at that changed objective function. So still just as strong, but we want to, oops. We've changed our objective function, which you may recall penalized high activations. We go down to our same muscles again, medial and lateral gastroc and soleus, and we go time and add them. You can see kind of a similar thing happened. We see reduced activation from all 3 of these muscles. Still reaching 1 for those 2 gastrocs, but not maxed out for nearly as long. So there's a lot in here in terms of, you could look at every muscle in this model as well as their forces, and I'm going to show that in my slides in the interest of time. But I would encourage you to dig into and change these parameters and plot how this affects other things in the model. I think that a big advantage of these types of models is that we can make a change to 1 muscle and see how all of the other muscles respond. And we can also look at a wide variety of perturbations in terms of these properties and see, well, how does this really affect our output solution? So I'm going to go ahead and stop sharing OpenSim. And then I am going to share my slides. All right. Are we back to slides? All right. So clearly there's lots of runs we could do, but this is basically what we saw. We saw these maxed out activations. If we go ahead and load the force files, this is what you would see. So greatest force output from the soleus muscle, lower forces from the gastroc, which are less strong. And certainly we're maxing out our activation. Is that model really strong enough? And then we added that strength model on top here. You can see at the force level, we saw that soleus went down and gastroc went up. So this shows that, we made our gastroc stronger. And so the agonist muscle didn't produce as much force. The gastroc produced some more. And then if we changed our objective function, we saw those activations go down more, and that affected that force output as well. So you can see the medial and lateral gastroc have higher forces, soleus went down. So we have more of an even spread of those forces across muscles. So a big question then is, we've run 3 scenarios, really quick and dirty, but with real data, we got 3 different solutions. How do we know which one is right? Does anyone have any ideas? How do we validate this? Again, unmute or chat. No wrong answers.
With our data, like EMGs.
Ding, 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 20 points for UCI neuromechanics. All right, so that's a huge one. And like I said at the beginning, anything and everything we can measure is going to help us validate. But EMG is a huge one. If you recall, when we ran these scenarios, we loaded in our kinematics, we loaded in our ground reaction forces, but we did not load in our EMG data, but we did collect it from this participant. So if we can compare those muscle activations to what we measured in the lab, that can build a lot of confidence in which solution might be right. And I think the big thing is, do we have an independent data stream, something we didn't use as input that we can then compare our output to? But there's so many things out there, and I think is a really exciting part of IMSI is, dynamic ultrasound, or potentially, linking species or linking models at other scales. Do we get a similar output from the spring mass models we just talked about, or potentially tissue level models that also predict activation and fiber velocity and forces? And do those match? How do we link these scales together? And I think another key take home, that if nothing else, this is what I want you to take away from the workshop today, is that in designing a project, it is so important that you have the modeling approach and the experimental approach designed together, because knowing what data to collect, what's really feasible, what your research question is going to affect what data streams you have available to you and what kind of models you can run. All right. That's me, and I'm not too far beyond time. I'm going to turn it over to Katie Knaus now. Katie, we can't hear you, or at least I can't.
All right. Can we hear and see my slides? Okay.
Yes.
All right. So I will be talking about kind of our next scale down in size, but maybe scaling up in complexity of our model and the data we need for it here, to talk about finite element modeling. So there we go. All right. So we've talked about these different scales of mathematical models, particularly thinking about models that describe the structure and function of skeletal muscle. And so Anne has kind of walked us through OpenSim, which is this whole body model that uses these models of muscle, hill type, or these kind of lump parameter models that represent the behavior of a whole muscle, but really kind of building from the behavior we know that comes from this microscale that we've talked about kind of a few workshops back, of what's happening at the filament level, and also in that kind of talked about models at this scale to really understand the mechanical interactions there. But with these kind of hill type lump parameter models, what we're assuming here is this idealized muscle architecture, this idealized structure. And so where I think muscle is really exciting is that we see very different structure and architecture across different muscles as they have different functions in the body. And so what I want to talk about today is kind of models that span this gap when we have questions related to kind of complex structure function relationship of muscles. So in particular, the kinds of models I work with are finite element models, which also utilize constitutive modeling. So we'll talk a bit about what those terms mean and how we use them in kind of a biomechanics and muscle context. So starting out, we'll go over kind of a very brief overview of finite element modeling, but in essence, we're talking about a mathematical approach that allows us to solve complex elasticity and structural problems. So widely used engineering tool. So we have this example of a problem here, a classic mechanics beam deformation problem. And so when we're kind of breaking down how we solve this problem, we are thinking through how we go from these external applied forces to stress within our material, the strain of that material, and how that results in deformation of our structure. In this case, kind of our rectangular beam here. So mechanics students might recognize this equation, which is relating delta or deformation or the extension of our beam to P, which was our applied load, and the length of the beam L, the cross-sectional area A, and then the elastic modulus E. And so this kind of comes into this progression of solving our problem. So we have boundary conditions, which is our kind of external loads conditions here. So in this case, our P, our applied load. Relating strain and deformation is the geometry of our problem. So in this case, with our simple beam, we have a length and cross-sectional area. And then we have our material properties, which is relating our stress to our strain. So in the case of our beam, this is a single parameter, our elastic modulus. And so kind of with these components, we can solve this problem. But if we weren't using kind of these simpler conditions, these simpler geometries, right, this would be a hard problem to solve by hand. And so finite element method is essentially discretizing our problem. So in this case, we could take our beam and divide it into all of these little elements, these triangles in the picture here. So where each element is kind of its own smaller mechanics problem, and then they have to agree with each other at their boundaries, right? And we treat each element as a continuum or kind of uniform properties, and we'll talk more about continuums in a bit. And so we can take a complex problem and separate it into these smaller problems, which becomes much more manageable when we use a computer. So not recommend solving these problems by hand, but with our computational software, we can simplify, solve many equations, and be able to address some complex structural problems. So how we apply this now in the context of muscle biomechanics. Similarly, we have geometry. We think about kind of our macro muscle scale. We're representing muscle, tendon, structure, and architecture, right? So when we think about our boundary conditions, we're kind of pulling from different scales for these different pieces of our problem. So thinking more from the body scale and our conditions of movement that are going to apply loads or displacements to our muscle. And then finally, material properties coming from that micro scale and thinking about the kind of properties of this muscle, its mechanics, both in kind of active and passive conditions. Right? And so the way we kind of define material properties and talk about them in kind of our finite element space is we work with constitutive models or constitutive equations, which is kind of a mathematical representation of the properties of a specific material. So if we think about muscle as a material, it's pretty complicated. We've got a lot going on with muscle, right? It's a composite of different things. We have multiple cell types involved. There's neural innervation that leads to active fibers. Cross-bridge mechanics play a role. We have nonlinear properties. Anisotropic, which means it has a preferred direction, right? We have fibers oriented in a specific direction. It has some rate-dependent behavior. We've talked about force velocity and some viscosity, rheological properties. So it's also incompressible. And so when we want to kind of define this within our simulation framework, we need a mathematical approach that we can kind of describe all of this physical behavior. And so in part, we kind of familiar with this force length or force stretch relationship, right? That can represent these both active and passive constitutive properties of our muscle. And so this is in fact the kind of constitutive relationship that shows up in our hill type models, right? We're mathematically representing this behavior. But for finite element modeling, we're representing the 3D behavior of muscle. And so our 3D models require kind of a more complex mathematical approach of our three-dimensional material or tissue behavior. And so this is where we use tensors and tensor equations. And so this is kind of widely used in materials mechanics, but it kind of on the newer end of biomechanics and how do we define constitutive laws? How do we define these tensor equations for our complex biological materials like muscle? And so this has been kind of started by the work of Y.C. Feng, who's considered grandfather of biomechanics in some contexts, so really kind of pioneer in this field of treating biological tissues as materials in these mathematical approaches so that we can define not only the alarm fiber behavior or the one-dimensional mechanical properties of muscle, but actually these three-dimensional behaviors so we can model three-dimensional materials. And so this kind of modeling approach, constitutive equations, relies on continuum mechanics. And so in continuum mechanics, we have an assumption of matter as a continuum. What that means is that for kind of a arbitrary unit of material, we assume it fills the entire space and it's infinitely divisible. So if we think about our steel material here and how we might kind of subdivide it, we would be able to go to a pretty small scale before our steel is kind of not filling the whole space. We can't divide it anymore, right? The level of kind of carbon and iron atoms. But at scales above that, we can assume just kind of a uniformity of steel, a continuous steel within our space, right? So this becomes a bit trickier with biological materials like muscle and tendon and many others, is that our scales, as we kind of move across hierarchy, are not quite as easy to make these continuum assumptions, right? We have to consider what scale of tissue we're working with to make these assumptions about it filling the entire space, being infinitely divisible. Because when we have a continuum, it's going to be defined with a specific bound of what our lowest size scale is. And so I'll pose this question in the chat, but when can muscle be a continuum? When is it okay to treat muscle like a continuum? So maybe when we're talking about a whole muscle or the tissue scale, kind of like closer to the fascicle scale, tissue more at the fiber scale, or even at the filament scale. I don't know that I can see the chat, but if you all have thoughts, drop them in. Maybe it'll pop up. So some answers. So, right, a lot of our answers in modeling is always, it depends, right? When we're working with whole muscle, probably depending on the size of our organism and its muscles, oops, we can isolate a continuum and kind of assume that the elements we're going to describe in our finite element model can capture muscle that is relatively uniform, continuous within our element, right? Fascicle scale, maybe we can have tissue with uniformity. At the scale of kind of fiber level tissue, we might have to separate our rather than kind of a continuous muscle material, like a muscle fiber material versus an ECM material, right? Whereas filaments, we're starting to move away where we can have this kind of filling the entire space assumption, infinitely divisible. That's where we move towards these more spatially explicit models like the ones we've worked with previously. So let's kind of consider our 3D modeling approach. So I'll talk about kind of 3D modeling in this case of the plantar flexors. So we just worked with the gastrocs and soleus and Anne's example, where we had kind of this 1D modeling approach from OpenSim. And now let's compare and contrast kind of what goes on if we have a 3D muscle model. And so to start, let's think about our simulation inputs. And for these two, we actually have pretty similar inputs here. We have activation, joint angles, function of time for whatever our task condition is. right? So the experimental data would be kind of what we need in this space. So welcome to type in, but I know we're short on time, so I'll kind of move through this. So we're looking at kind of similar data to what we described previously for the OpenSim model, right? This kinematic data, gravity reaction forces, muscle activity. But then for the 3D model, we need some additional experimental data to actually build out this model, right? So people mentioned muscle properties. And so to build a 3D model, we usually need pretty detailed muscle shape, architecture data, used typically from imaging approaches. All right. And then, so they have similar inputs, but when we look at the simulation outputs, this is where we start to see some differences. So similarly, both these models can predict muscle forces. They can contribute to how we might predict joint forces or muscle tendon unit lengths. But with our 1D or OpenSim version of muscle, right, we're looking at kind of a single fiber length, a moment arm for the muscle tendon unit, a single tendon strain parameter, where in our 3D model, now we can look at fiber lengths individually. So how these might vary through the muscle, how each might kind of contribute to mechanical advantage. So moment arms of specific fibers and other tissue stresses and strains. So rather than a single tendon strain, we could look at where we see strains in the tendon, the aponeurosis muscle itself. Right. And so while these models give us a lot of insight, they are quite complicated and come with trade-offs. So when we look at our computation time, our 1D model is much faster than the parameters we need to build a 3D model. We kind of have fewer that are known, right? So we don't have, we have more parameters that we need and more data we need to collect to build it. Further, the assumptions we make in these models are less established, kind of working on building these up and more of these, but still they have not been used as long traditionally. So we just have kind of less of our establishment, both of the assumptions that go into these models, as well as our validation approaches. So we talked about validation practices for a 1D model, and similarly with the 3D model, we would validate, but kind of less standard practices and more questions about what experimental measurements might be the best to approach this. But 3D models really do have some advantages for specific research questions. So if we are concerned about a particular quantity that is kind of simplified in the 1D model, a 3D model would be a chance to examine that. So, or kind of capturing specific feature or effect, or predicting a quantity that the 1D model can't predict. And so I'll go through kind of a very brief example, the soleus muscle. So this is a very complex muscle that we have studied with 3D modeling. If you're unfamiliar, it has multiple compartments of fascicles with different architecture and this kind of complex interdigitating aponeurosis structure. This is the human soleus, to be specific. And then, so if we kind of think of our hill type model, right, there's some complications in this soleus architecture that are not really captured by this lumped tendon aponeurosis structure, this uniform muscle fascicle assumption. And so this is our model of the soleus. So this is the soleus lengthening through dorsiflexion. And what we see here is kind of predicting strain through the muscle during this motion. And so one of our questions here of what data would we need to kind of set up this model to build the simulation? Toss those in the chat. Of course, we can. Idea, yeah. So we kind of have twofold needs in building this model. The first is actually creating the model itself, the geometry, the structure of it. right? And so this is coming from detailed shape and fiber measurements that come from things like high resolution MRI or fascicle reconstruction from DTI, diffusion tensor imaging, different MR technique, and then, or even cadaver dissection to kind of really understand this complex structure of this muscle. And so that's to build the model itself. And then we have to create simulations, right? And this is where it kind of links back to, it's more similar to our 1D model, where we need our kind of motion data to know how the muscle is changing length and how it's activated over time. So kind of on the validation side, some guesses of what we need here to validate a model like this. So a challenge in 3D models is there's a lot of degrees of freedom in the model. So validating, we kind of need a wealth of data to examine different things. A big factor is using imaging, particularly dynamic approaches to imaging, right? Because we're predicting with our simulations kind of dynamic muscle function deformation. So things like dynamic MRI, where we can actually capture muscle deformation in vivo, or ultrasound. Some static MRI techniques where we can get kind of higher resolution images like DTI, but are not actually able to do so while the muscle is active and moving. So we have trade-offs between kind of the level of detail versus what is kind of capturing true dynamic in vivo conditions. So our dynamic MRI, we're limited to kind of whatever range of motion we can do within our MR scanner. Ultrasound, we can capture kind of more complex dynamic conditions, more similar to kind of physiologic movement. But again, we're kind of sacrificing some of the complexity of what we can capture, right? We're looking at 2D planes. And then, so this tells us a lot about deformation, and we can validate that our model shape changes and strains look correct. To kind of further look at stresses and forces, we would need kind of further measurements, so estimates of forces if we're working in vivo with humans, which was the case with this model. If we're modeling animals, sometimes we have more direct measures of muscle forces that we can work with. All right, so just kind of wrap up with some questions that we can answer with this kind of model. So with this Soleus model, we looked at how fascicle behavior actually varies across the muscle. So rather than that assumption of a single fascicle behavior, we can look at across the muscle within these different compartments. Further look at how the activation dynamics affect fascicle behavior. So the top graph is passive stretch of the same muscle extension versus an eccentric contraction of the same muscle extension. And we see kind of differences in those fascicle behaviors. And then kind of look at interactions between activation and length, especially to think about how it's affecting stiffness at an organ tissue scale. How the aponeurosis properties, so I mentioned these, they kind of large aponeurosis structures interact with the muscle tissue, and so what role those properties play in muscle fascicle behavior. And further, kind of how these variable mechanics of the soleus play a role in different functional tasks. So can't go into much of these today, but we'll be talking about it at ASB if anyone's there. So this is kind of an example of a very detailed model where we had specific questions we wanted to answer. But I think kind of wrap up with this thought is that our required experimental data is really going to depend on what type of model we're using. And then the type of model needed is really going to depend on our research question, right? So the level of complexity needed is going to come back to that question. And so we'll end with a chance to ask questions, and please give us your feedback on this workshop. So the survey is here. You can use the QR code, or there's a link in the slides.
Anybody have questions?
I just want to say this is such a wonderfully full of information workshop. I'm definitely going to want to rewatch the videos. I know you push out the videos for all of these things. Are they all in the same place, or do you the individual links for each one?
Everything is shared. There's one workshop folder that has subfolders for all of the workshops. The link for that folder is in the curriculum document that's pinned to the Slack channel. And then we're saving all of the recordings to the individual folders with the materials for each workshop.
Fantastic, because I'm definitely going to go back and catch even more of this. Thank you.
Monica, I have a question for you, which is that
Yeah.
your predicted ground reaction forces are so beautiful and smooth. And I'm wondering if you can comment on the high frequency components we often observe experimentally.
How many frequency, what are the frequency components we observe experimentally?
No, just the fact that
Or
there
just
are
the fact that there
different
are
ones, right? And
Yeah.
how we might predict those.
Yeah, that's an interesting question. So the model, the mass spring model, the simplest mass spring model has no mass, it has no foot mass, it has no leg mass, right? And so if you developed a more complex model that had a foot mass or multiple segment masses, right? Then this would lead to potentially some higher frequency components as each of those segments is decelerating, right? When the foot comes in contact with the ground. But another thing that is not included in models often is that there are large soft tissue masses, right, on the bodies. And they are not rigidly attached to the skeleton, right? So there are bouncing motions of the soft tissues of the bodies when the feet come into contact with the ground. And some of those higher frequency components could be associated with those soft tissue motions as well.
Hello, do we have time for more questions? We've got one from Tech.
Sure. Yeah.
Mainly for Katie. So when I see the color maps of your muscle, I freak out a little bit about things like hill type models. And so I'm wondering, like, do you have thoughts on how to inform a modified hill type model, or how to inform modified lumped muscle models, given what you have shown us about the detailed strain and stress patterns within the tissues?
Yeah, so I think kind of multiple answers to that, right? So things we're kind of thinking about is how to best represent the like tendon structure of a hill type model, particularly in these muscles like the soleus, gastrocs with the kind of significant aponeurosis portion. And so trying to better understand how we can kind of characterize those lumped series elastic components of those models from what we can learn from the imaging and the modeling of those parts. I think another kind of question is where to kind of take our average muscle measurements, right? So our hill type relies on a single kind of fiber characteristic, right? So fiber kinematics are, you're going to have the same length changes, pronation changes. And so with these kind of multi-compartment muscles, thinking about the variability across the muscle and kind of what does it mean if we're kind of only sampling in that muscle belly as far as what we predict the behavior of that muscle is versus kind of what's happening more at the extreme ends or the deeper compartments. And so that's kind of, yeah, questions I have is like, how does that change our hill type model if we kind of make different assumptions about which part of the muscle we are calling our idealized muscle behavior? So I don't have like a straightforward answer right now of like, oh, we should definitely change our hill type models to this, but more of like, what are things we can consider? So as we do ultrasound collections, right, can we measure in different regions of the muscle at different times or with a dual probe so that we can start to kind of understand those assumptions of when we take kind of an isolated field of view of what a muscle is doing.
Yeah, I agree. Like the amount of strain that you get sort of distally versus in the belly is so different. And that, I mean, if you think about what the sarcomeres are feeling and what cross bridges are doing in those different places, it's really not the same thing everywhere, right? So,
right.
You know,
yeah, and I think
for generating cool data.
Yeah.
Make us all work harder.
Yeah, I think there's a lot of open questions like what the sarcomeres are doing. So from like an adaptation point of view, are there kind of regional differences? I think from an energetics point of view, right, we kind of base it on our fiber behavior. So what does that mean for a whole muscle that has variable fiber behavior? So yeah, definitely. And I guess introduces more questions maybe than we've answered them yet, but certainly fun.
Katie, there's also a question for you in the chat from Kisa. How do you get the different parameters for active and passive muscle in the simulations?
Yeah, so I guess parameters, so material properties wise, a lot of that actually comes more from like animal mechanical testing, but then trying to compare to dynamic imaging of humans to kind of validate those assumptions, right? Because we don't have great direct mechanical measurements of human muscle. And then kind of active parameters, similar kind of our active force length curve and fitting those, but then for our kind of activations themselves is still kind of an open question, right? With OpenSim, we can apply kind of a single activation per muscle. With the finite element model, we have some option to kind of vary activation through the muscle. So the simulations I showed, we assumed like a uniform activation across our whole muscle, but like things we've worked with is varied activation in different compartments and how that changes the muscle. So part of it, we kind of rely on the measurements we do have. So like the electromyography, we're usually measuring, again, kind of like with ultrasound, kind of a single location measurement. And so maybe don't always have great answers of whether it's variable. So a lot of what we do with the models sometimes is just kind of these what if scenarios of like, if we vary this regionally, does it matter? What role does it play? How sensitive are our outcomes to that when we don't have data to define those parameters specifically?
Are there specific parameters that the model results seem to be more sensitive to from those what if experiments?
Yeah, so a big one I think is kind of interesting is the aponeurosis properties, because it really kind of creates these bounds on the muscle fascicles. And so I think more so than we think about tendon in series with muscle, right? But when we have aponeuroses that are kind of in the middle of our muscle and wide, flat surfaces and strange shapes, it's definitely not an in-series relationship with fascicles. And so there's some kind of interesting mechanical consequences of changing aponeuroses properties that are kind of less intuitive and yeah, can make a big difference of how the fascicles behave.
Any other questions? I know we're going a little bit over time, so people obviously they can leave if they have to, move on to other things. But if we do have questions, I don't want to cut off the discussion either. So we have a question in the chat from Leah.
And says applied force velocity parameters under the static optimization. That's definitely true. So the force velocity, so in some static optimization formulations, you can have a very simple activation to force relationship where it's basically just activation times maximum isometric force equals the dynamic force of the model. And by checking that box in the static optimization, where we are electing to use the force length velocity relationships that are included in the Hill model to develop a better estimation of muscle force. So it affects that muscle recruitment solution.
Gotcha, thank you. I was just curious because I know static is, something not moving. And I was like, but we're jumping here. But it's just the idea or the pattern from the force velocity curve. That's okay.
Definitely.
Thank you so much.
So one of the reasons I chose static optimization here for today is because it is much faster to evaluate. So it's something we could feasibly do in a half an hour time, but it definitely has its limitations where each time step is evaluated independently of the others. So you can incorporate calculations of velocity in that prediction, but we're not necessarily making sure that we get a solution that is continuous from one time step to another. And you're right,
Okay.
for jumping, maybe static optimization isn't the best choice, but could provide us with an initial guess or an approximate of what might be going on.
Amazing. Thank you so much. That your presentation, all your presentations were so clear. For someone that doesn't have a modeling or engineering background, I was like, oh, it's making sense. So thank you guys so much. And I noticed there was like, the pattern of what do we put on the input, the output, how do we validate? I love that. and it really like helped with the take-home messages. So thank you guys.
Wow, what a compliment.
Thank you. Yes. We love your enthusiasm. Thank you so much. We'll go ahead and sign off for now. Thanks everybody for staying a little bit longer for the discussion. I will post this recording as soon as Zoom manages to process it. So thanks everybody, and we'll see you again next week. Bye.